Okay, I guess we should go on in the interest of time. So I'll be repeating some of the points that Laura made, but I'll try to be more concise and skipping over a few slides just because we are running a bit late and shuttle does leave at 6 o'clock. So I'm going to start by presenting some historical perspective on the solar as it was developed. Then I'll sort of gloss over downloading and installing it as a package. Then I'll talk about creating solar analysis directory and going about common analysis types, including univariate and multivariate analysis of variance, linkage, and association. I'll spend a bit more time on automating solar processing and how to perform those analyses in parallel computing environments, which is something that we must do given a large number of traits that we all deal with. And I'll be, unfortunately, Tom Nichols couldn't be here because he was working on the part describing the correction for multiple comparisons, but hopefully next year. And then I'll talk where solar is hopefully headed. So solar is a proud child of very esteemed parents. It was developed by as an offspring of three classical genetic analysis tools, which are MENDEL, FISHER, and SEARCH. I mean, those tools have been developed actually in the 1960s, where the quantitative genetics took hold, and as Laura mentioned, they were actually developed originally for the agricultural needs to stratify the milking cows' pedigrees and such, and then converted to quantitative genetics of human traits to deal with Mendelian disorders. So Mendel was developed for both clinical and agricultural geneticists for performing gene locus mapping, pedigree segregation, multi-point linkage quantitative trait mapping, allele frequency estimation, paternity testing, as well as genetic counseling. It came with two modules for counseling for cystic fibrosis and specific type of dystrophy, and then macular degeneration and something else, all developed for actually genetic counseling performed in the clinics back in the 1970s and 60s and 80s. FISHER is a genetic analysis tool for quantitative traits whose variability can be explained by polygenic inheritance as well as environmental forces. So you can kind of sort of see the polygenic command. The polygenic command in solar performs the analysis that originally were developed as a FISHER tool. And it provided functions for analysis of continuous polygenic traits, such as blood pressure and, interestingly, total left and right finger reach, arch, and look count, which apparently was a very hot polygenic trait that's influenced by 7 genes back in the 1960s and 70s. And obviously it has strong environmental influences. And then both of those packages used variance component analysis, where the variability in a trait is either multivariate normal or t-distributed, and supported both multivariate and univariate analysis, including proband correction and robust outlier detection, and used maximum likelihood estimation engine provided by the SEARCH tool. So SEARCH is the original maximum likelihood estimator engine for Mendel and Fisher. This is a very classical and very powerful optimization routine which worked with user set bounds and limits and maximizes a given cost function. So it samples a function over a grid and cost functions can be implemented as either least square or user-defined cost estimators. And I think John Blangero is the one who optimized the search routine for discrete traits, and the new name was given as Optima. And as I understand, in the most recent version of Solar, the search algorithm has been actually replaced with eigenvalue decomposition estimation, and that's what Laura can correct me if I'm dreadfully wrong on that.
I don't believe that's in the version that's available on the website yet, but that will greatly speed up calculations in large pedigrees.
It actually is available on the website, yes, and so if there's going to be a link that's going to be flashing several times over the next few slides where you're going to see the link, a place to download it. So conceptually, in terms of software architecture, what is Solar? I think it's kind of important for us as people who deal with methods and tools to actually understand the software architecture in order to better use this tool. So Solar is composed of TCL interpreter that sits on the top level. Oh well, my laser pointer doesn't work. And so the TCL interpreter takes care of all the Solar functions used by the high order processing. And so all the functions that you actually type in the command prompt are procedures or functions that are written in a TCL language. And TCL stands for Tool Command Language. So TCL also codes for the Fisher and for the parts of the Fisher and Mendel algorithms where but then it calls high performance C code that sort of wrapped around with TCL layers. So when you actually type Solar, it's the TCL interpreter that starts, and you're now working with it like with an interpreter, very similar to the way R package works. And when you type a command like polygenic, the TCL interpreter reads solar.tcl file, which actually codes all the popular functions, including polygenic, and executes the polygenic function, which analyzes the arguments and existing global variables and chooses among several analysis types, and then calls upon objective libraries for computationally intensive procedures. So TCL is a tool command language. It was a script language that was developed in late 80s. And it's very similar to the shell scripts such as Bash and SH. And essentially you're working in a TCL shell. And as I said, all Solar commands are TCL functions. So you can pre-use, copy, or modify any of the commands by basically typing either showproc and the name of the command, such as polygenic, or you can run it this way, showproc command underscore polygenic my command.tcl, and it will actually create a TCL script in your directory and copy the original TCL script into that file, which is very convenient if you want to modify a specific function that you feel like needs some modification for one reason or another, such as performance optimization. Again, you can download and install Solar. I give you the Nitric website where you can actually download it as all packaged up, including the PowerPoint presentations for today's meeting. And you have the one version that we actually put on that website is the beta Linux version, which has the EVD feature. And this is actually, I think that's the only version that can run GWAS scripts at much improved efficiency. And then you just email Charles Peterson for free registration code, and then you can pre-use the manual at the website that Laura already showed you. So Solar projects are organized as directories, and that is not to say that you cannot run Solar processing on data that's outside the directory, but Solar sort of assumes that all the data and all the pedigree files are located in the directory from where it's run. So upon the startup, Solar reads the default files from the directory it started from, and that includes both pedigree and default phenotype files. And directory should either contain the pedigree file in the PEDSys format or CVS. And I think Laura has really went over with it and explained how those are created. So once you loaded your pedigree, you can actually look at the pedigree file, and this information may be of some interest to you if you are interested, if your interests are involved more deep understanding of the pedigree effects, such as if you want to do permutation or something similar. Same goes for the pedigree structure. So you just some explanations of things that you might want to look into if you're actually using Solar for a complex project. And for Phi2 file, I think Laura has given us really good explanation of what is what. So Solar is quite a popular package for genetics people, and for conversion in and out Pedindex format is implemented in several genetic software packages. So if you work with a geneticist that uses some form of genetic program in his or her research, his or her genetic program might actually read Solar format or convert to Solar format. So, for example, one of those packages is library Multic, which is available as R library. So, given that most of imaging research at the moment involved GWAS, coding the matrix, pedigree matrix for unrelated subjects is actually extremely easy. You can do it in about 5 minutes with a simple Excel file and then save it as a comma separated values. So basically make a text file or in Excel, well, basically create an Excel file where you load all your subject information, and then, as Laura said, and as this slide shows, just go ahead and code it using your own ID and then put 1 or 2 for male, female. The only advice is don't end your lines with commas, because that can actually create a problem. So PEDSys program, which is also a program available for free download from the Texas Biomedical Institute, can convert complex pedigree files from multiple other software packages, including those listed and others. So if you already have some statistical software that this pedigree exists in, there is a good chance that you'll be able to simply convert it to the Solar format. And again, coding your pedigree manually can actually be done fairly easily, as long as your pedigree does not involve multiple generation. So, coding your data set that's family based, where you only look at mother, father, and their offsprings, can actually be done in Excel without much difficulty. We've done it before, at least. Now, I think Laura has already demonstrated how S&P file can be created. Well, you can also make it in her presentation. She coded the SNPs based on their allelic names. You can actually code it directly using 0, 1, and 2 format. The only important part is to make sure that you have S&P prefix for each of your SNP names. So this way the GWAS function will actually know that that particular trait is a SNP rather than a phenotype.
So, Peter, can I comment on
Yes.
this a bit? If you do it the way I was showing, where you load the genotypes and then use the SNP crowbar command to make those 0, 1, 2s, it will append the SNP prefix for you. If you're loading them already as 012s, then you need to have that SNP prefix in the names in your file.
Yes, very important. And then map and frequency files are needed for QTL processing. And I actually, in the interest of time, actually advise you to look at a fairly complete instruction page on the website explaining how this is done. It's actually not all that difficult at all. Now let's talk about phenotype files. Again, the only data format that Solar supports when it comes to the phenotype files is a comma-separated value format. So it's a simple text file where individual values are separated by commas. I mean, it also supports PEDSys phenotype files, but you're unlikely to encounter it in your own research. A header must include either ID or EGO field, because that identifies it as an ID field. There could be difficulty with Excel if your first column is called ID because it assumes that it's some obscure data format and then might actually refuse to load it. Yeah, it's a known Excel bug. But you can actually move that ID field to your second field or third column or fourth column. It doesn't really matter for the Solar purposes, but just so you know. And typically ID is used for human subjects and EGO is used for animal subjects, but for your purpose it doesn't make a whole lot of differences. So your typical file will look very much like this, where you have ID. So the first line is considered to be a header, and then you go, which includes ID, age, sex, gray matter thickness, fractional anisotropy, flare volume. And then you go by ID, then age, sex, and so on and so forth. So pretty simple format. Now, given that Solar can only read CSV file, implies that we cannot expect it to read binary imaging data. And why is that? Is because TCL is a very simple interpretive script language created back in 1980s. So it's not designed for handling large chunks of memory typically associated with binary data. It also does not support multidimensional arrays. In fact, it hardly supports unidimensional arrays. And in Solar, all large files are actually not handled by TCL, but directly passed to the underlying, high-performance programs which handle that type of processing. And therefore, at this stage, the preparation of imaging-based phenotypes is left to us as, the end users. So it's now up to us to convert the binary data to the CSV format. So how do you go about preparing your voxel-wise traits? Because, that's going to be the most difficult case to deal with. So it can be done by two ways. It's useful to save your imaging data in a 4D Nifty file, or you can actually directly have a directory full of the imaging files and just include the genetic ID in the name of the individual files. And then you'll have to write a program or a script or MATLAB script that will save voxel-wise values in the CSV format. So Solar has a limit of 40,000 traits, 40,000 traits per CSV file. In my experience, and I strongly advise it, it's not to exceed 10,000 traits per file, because TCL can be dreadfully slow when it comes to manipulation with memories. So TCL doesn't really support even one-dimensional arrays. So what it does, it actually makes a new variable for each of those values. So therefore, you're actually creating 40,000 new variables in a TCL, and it's really not designed for that. So typically, multiple files are required for voxel-wise studies. But this also presents us with opportunity to fit the topology of a computational cluster. So if you create your phenotype files based on the number of cores you have in your cluster, that will sort of help makes it more logical when it comes to the actual processing. So let's assume the simplest case. So you have 300, 400, 500, 600 files, and all of those image files are now labeled as MRI ID, genetic ID, sex, and gender. I made a really small C++ program that you can download from this package, and if you really actually want to compile it, you can download the necessary libraries from the link below it. And this simple C++ program uses a mask file, and it creates CSV files and a header file for each of those files. And each trait now is labeled as voxel XYZ something. So you're basically going to have a large CSV file with columns labeled voxel 128, 118, 10, for instance. And typically, I attempt to save maybe between 1,000 and 5,000 traits per file. So you ended up creating about 200 to 300 files by that program. Oh, I forgot to mention that this program is distributed as Eclipse project. So if you use Eclipse development environment, you'll be able to basically just plug it into your development library, and it should work from there. If you don't use Eclipse, you can just take the main.cpp file from it and pre-use it and attempt to recreate it in MATLAB or whatever other development environment you like to use. So now that you have created your phenotype files, how do you go about performing the analysis? Well, as Laura said, you basically start SOLAR by typing SOLAR. And you read your phenotype files by typing, load pheno and then name of the file. And assign a trait by typing, trait voxel 111. And better yet, you inverse Gaussian normalize it first using the same inorm function that Laura was talking about. And that actually provides a pretty damn good insurance for the normality of the trait. Then you assign your covariates, and all analysis I typically perform covariate for age, h square, sex, h and h by sex and h square by sex interaction, and that line can actually be compactly coded by the line you see at the very bottom of the slide. So you type to perform univariate analysis of variance, you basically type polygenic, and it comes up with a pretty long list of symbols and numbers, and this works really great for when you're analyzing a single trait. This can be a bit overwhelming when you actually analyze 500,000 traits. In addition to that output, a directory which actually named after the trait will be created, and it contains several files, some of which can be useful. So it contains .mod and .out files, and typically .mod files contain model parameters. So for example, null0.mod contains the zero model, including the results for each iteration for the MLE processing, and same goes for the sporadic and polygenic models. And polygenic.residual file typically contains the residual values after correcting for all of the covariates. And file polygenic.out contains the copy of the results. Now, when it comes to processing, 10,000, 20,000, or 500,000 traits, I suggest that you use SE_univariate_polygenic script. This is the script that I typically use for my processing, and all you have to do to use it is download it from the link above and then place it in your working directory. It's a TCL file, so you can actually, go over it and see what has been changed. And what it does, it's so do everything the same that you've done before doing polygenic, and then type SE univariate polygenic instead of polygenic, because it makes for about 10 to 20% quicker processing time, and it restricts writing many of the intermediate results, which saves both time and disk space. And the results are now saved in a tab-separated file, which makes it for a lot simpler follow-up reading of the results for 400 or 500,000 traits that you've just processed. And the name of this file is SE_out.out file, and you can convert it back to the image format by a program called assemble univariate_image. And it actually reads a mask and that SE_out file, and it creates three binary images for you, which contain heritability, probability, and percentage of variance explained by the covariates. And that's, of course, for voxel-wise values. Now, where I believe SOLAR really shines is the ability to perform multivariate analysis. As Anita was asking about what happens when you have a phenotype that is strongly influenced by age. Well, by actually using age as a covariate, you do indeed lose. You can potentially lose a lot of your genetic power in the analysis. So one way you can go about it is to perform bivariate genetic analysis. So if you have two traits that are equally influenced by trait, then by performing the multivariate genetic analysis, you can restore this power. Don't ask me how it works. All I know is that there was a very interesting, there was a very dry statistical paper from Houston back in, late 90s, which actually did a lot of simulation type analysis. And they demonstrated that by using bivariate approaches, you can get between, 10 to 100 power improvement in the power of genetic discovery depending on your pedigree structure. So I think that is sort of for real.
And depending on the type of pleiotropy.
Right. So the idea is to choose two traits which you believe are pleiotropically influenced by the same genetic factors. And by using bivariate analysis, you can now avoid the unpleasantness of losing power when you covariate age out. Or so it was explained in the paper.
I have a question. I always run into this kind of, I have this multivariate approach. So when you suppose you have a gene, influence the variation, but then you do is the touch circuitry, which should correlate. But when you do the multivariate analysis, and this trait correlates, but statistically you are trying to put into the multivariate, you have to work around a multipolarity problem.
No, actually you don't. Because what happens is you're analyzing shared variability between those two traits or multiple traits.
So when I'm trying to use the circuitry, and the circuitry, of course, will go back and highly correlative.
Right.
I don't have to vary statistically the multipolarity.
No. So you actually want it to be there, because what you actually do is you're not analyzing variance in any of the individual traits. What you're analyzing is a genetic influences on the variance that is shared among two or more traits. So that's what you're actually getting. So it's not like a factor analysis where you're trying to distill and orthogonalize, variance across multiple axes. No, here you actually try to find variance that is shared between several traits, and then find genetical factors that influence it.
But the models help you the most. You get the best increase in power when the second trait is providing some sort of additional information. So that paper that Peter referred to, I think was by Chris Amos and colleagues, and showed that you get the most bang for your buck when that genetic correlation and the environmental correlation are in opposite directions, where you've got traits that are positively correlated genetically and negatively correlated environmentally. And that sort of structure gives you the best increase in power. But even if you haven't got that, if you have this genetic correlation, if you have either the genetic correlation or the environmental correlation even, you can get an increase in power. But if you have two traits that are essentially identical, they're essentially measures of the same thing, then you might not get an increase in power out of this because they're not actually giving you any independent information.
So was that paper, was that imaging genetics paper?
No.
It's a statistical paper that was tried.
Yeah, but so presumably the issue then is you can imagine that if you have two voxels, so it sounds like the reason that method is going to work well is because you're getting rid of noise. You're looking for shared variance, which you think is more representative of signal.
Yes.
Less representative of noise.
Yes.
So then based on that with imaging data, the more your voxels are likely to share noise, the less benefit there will be. So presumably it's going to be most helpful if you're either using data from different sessions or from voxels which aren't that close by, where there's less likelihood of shared noise.
Yes, so where you have some hypothesis that there should be overlapping genetic sources underlying them.
Yeah.
And the scenario I would see as most useful is, for example, you're probably talking about Alzheimer's disease, ADNI data. So one potential scenario where I can see this to be very helpful is when you use, for example, brain imaging data, and then data from blood or CSF-based markers of genetic activity or proteinomic activity.
You could also imagine if you had PET data and anatomic data.
So actually, this is
Right.
I was not clear,
That was
but
what
when
I
I
was
was
guessing.
talking about multicollinear trait, I was thinking of the imaging data. So when you are doing just a multivariate approach and you are looking for genes which related to all these traits, the brain region or activity or whatever you measure as a quantitative trait, what I was told or told, I have to care of in the multivariate when I'm putting together this brain region, that multicollinearity problem. So what you are saying, I don't have to worry about when I am choosing the brain region is actually better if they're correlated.
Yeah, because
So you
it's a different
I
issue.
clear?
It's a different issue. You're making an assertion about the multicollinearity, but you're making an assertion about the multicollinearity in the phenotypic space.
Yes.
And what he's talking about is the relationship between the genetic and the environmental space, which may or may not have that same
So it's a
issue
two
of.
different issue.
Yeah.
So go back.
I don't think anyone has. I've never looked at issues of collinearity in genetic space. Although what Laura's saying is that it may be less informative,
Okay.
which would make sense. But since Mike Neal lives in this space, maybe you should ask him.
You're assuming I was paying attention.
I think there's questions here of does it cause computational problems and will it run? To which the answer is no. And there's questions here of
What does it mean?
is it a useful study design to use?
Yes.
If things are really collinear, there's almost no point in doing the multivariate analysis
Yeah.
because the measures are essentially measuring the same thing.
Yeah.
So there is a sort of substantive question about whether it's worth going to the trouble of multivariate analysis if the measures are extremely highly correlated.
It'll run, but you may
Right, but the question is also the thing that Laura's put forth is that just because if it's not extremely highly correlated phenotypically, so it's not like 0.999, some sort of thing, right? So let's say you have something that's like a 0.7, right? Which in a general sense you would say there is a lot of collinearity there, so you're not seeing a lot of independence. Yet when you deconstruct it into a genetic correlation and an environmental correlation, it could be actually rather distinctly different, right? So there is a potential for you to have some benefit in that regard. Except I don't know how to screen that beforehand.
That's the issue.
Yeah.
Imaging data.
Yeah, I don't know how, I have no sense of, I have no feeling for that.
Well, let's carry on. So, for doing multivariate analysis, and I'm presenting this as bivariate analysis, you can actually add, 2, 3, or, as many traits as you want, as long as, you have enough statistical power to carry on with it. You basically define 2 or more traits by the same definition you define your single trait. And then you execute polygenic command. And in addition to the polygenic command, it's now advisable that you include 3 magic keywords, which are test raw E, test raw G, and then test raw P. What this will do is it will calculate phenotypic correlation, which is basically Pearson R, and in our notation it's going to be known as raw P, and then decompose it into 2 Pearson correlation coefficients, and that's Pearson genetic raw G and environmental Pearson environmental raw E parts. And here you can actually see the relationship between them given the heritability and so for this sort of analysis you actually do need related individuals in your pedigree, and it can either be families or twin siblings. And then you can actually perform the analysis like this. And once you set up your traits and covariates and executed the analysis, you will get a pretty lengthy report on all the results of the processing, including raw P, which stands for the phenotypic correlation, or your typical Pearson correlation coefficient, which is, by the way, corrected for relationship in your pedigree, so it's sort of like a mixed model approach. Then you get raw G, which stands for Pearson genetic correlation coefficient, and then raw E, which stands for Pearson environmental correlation coefficient, which is a correlation among residual environmental variants that could not be accounted by existing covariates. Now, for much faster calculation, you can use SE_bivariate polygenic script. And again, just get it from that package from the Nitric website, place it in your working directory, set up your traits, covariates, and type the SE_bivar polygenic. So there's going to be no need for any extra parameters, and it will calculate all 3 Pearson R's as default. And results are now stored in the SE_bivariate_out.out file, and their tab separated values for correlation coefficients, their standard error, and their significance. Now, QTL analysis is something that you guys are unlikely to experience, but I included a couple of slides just in case there are people who are interested in this, and we might want to expand upon this part because it's actually quite interesting and quite brilliant, frankly, analysis. So QTL analysis identify chromosomal regions that are co-inherited with the trait. So you do need related individuals for those kind of analysis. And then you need chromosomal information, which now can be either gathered from standard chromosomal marker files, or you can actually use genotype S&P marker files. The latest version of SOLAR is supposed to be able to perform QTL analysis based on the genotype standard 1 million genotype markers. And the location for the chromosomal markers are stored in the files known as middr.info in the processing directory. And a lot more information on how to set this up can be found in the corresponding manual pages. For the QTL analysis, you basically choose your traits and covariates. And you can do it both univariate or bivariate or or multivariate way. So basically you type trait command following by the number of traits. Then you have to create a null model, which can be done by command polygenic. And then you set up your chromosomal range and scan intervals and execute the QTL analysis by typing multipoint. It does take a bit of time, so I'd say it can take as long as 1 hour per QTL analysis, but it can be highly informative. So the results are stored in a multipoint 1.out file and include chromosomal location and as well as logarithm of odds. So anything that's logarithm of odds where the logarithm of odds is greater than 3 can now be considered as significant evidence for linkage. And there is a summary of the results that is stored in the multipoint.out file, which also gives you where the highest LOD was. So there are several ways you can actually perform SNP association analysis. One of the simplest ways is to treat SNPs as covariates, and I think John Blangero is the one that suggested that to me. So the significance of SNP as a covariate can now be used as the evidence for association. So that's for quick and dirty sort of way. Well, it's not actually all that quick if you want to do it 1,000 times, but if you want to do it a couple of times, you can do it really quickly. Or you can use MG association function. So it only works with the latest Linux version of Solar, but the Mac version, as I understand, is coming very soon. And this function, processes all SNP traits loaded at the moment and saves the results in the file that Laura was talking about. Another cool feature about Solar is you can actually now do bivariate GWAS analysis. So where this comes important is that now you can see if a shared variability between two or more traits can now is influenced by a particular allelic frequency. And you'll have to use a command called se_bivmg, and this function was developed by Jack Kent. And what you do is you set up your covariates, and then you run the function using trait 1, trait 2, and give the the particular allelic name or particular SNP name, which will estimate the model with and without SNP, and then significance is calculated based on the ratio of odds. And results are now stored for each SNP. They're stored in trait1_trait2_rs text file, which has chi-square values for both traits, then chi-square for first and the second trait, sample size, rho g, and other information. So this sort of analysis can be done on both related and unrelated individuals. So actually, if you want to do a multivariate GWAS analysis, you can do it using this function. I don't see why not.
But I don't think that's what he's actually talking about.
He's talking about multiple traits, but yes, there's no reason you couldn't make static interaction sorts of models by specifying your own covariate.
As a trait. So now basically instead of your phenotype, you use basically a genotype as a phenotype, and then looking at it this way. Now, the main question comes on how to automate solar processing. So the right way, which John and Laura would advocate, is to write property CL functions for looping over the set of traits that you have in your memory, and that greatly capitalizes on the flexibility of the solar design. So the completely wrong but still usable way is by overwriting .solar file. So .solar file will be executed by Solar the moment it actually starts. And I've done it before, where you can basically create a new processing directory, then you create a .solar file in that directory, and you fill it with all sorts of commands, and then you basically execute an instance of Solar in that directory. Again, this is highly not recommended because Solar can attempt to read this file multiple times, and this can cause unpleasant behaviors. And then there is an easy way. So the easy way that again, you can do it is basically create a text file where you type all the Solar commands that you think you're going to need. So for example, create a text file named make_my_processing.txt, and then you use Unix redirection mechanisms by basically redirecting that file into the Solar. And because Solar is an interpreter, it will start reading that file line by line and executing it line by line. So you can use this for scripting your mass univariate, mass bivariate, and mass QTL processing. So I've made a few shell scripts that will actually create the massively, those Solar command files. So for example, a file that you'll find in that package called run make solar univariate file underscore inorm, it will take the header file and the trait file and then generate a .run inorm file, which will contain all the Solar commands for this processing. So for example, if you have, it's very convenient when you start, running into the number of traits that, exceed 5. So for example, if you want to run a voxel-wise approach, this basically becomes inevitable to use something like this, whereby you prepare a Solar command file for each of your phenotype files, which can now have maybe 4 to 5 to 10,000 individual traits. And this is in fact a very simple shell script, and you can look over it and modify it to your liking. And the simple text file that it produces basically looks like this. It's basically a concatenation or a sequence of Solar commands that you execute one after another, after another, after another, and finally you forward it to the Solar interpreter and it will run it. Similar concept has been used for the bivariate analysis scripts, where you combine all traits in a single CSV file, then you create 2 text files with the traits on both sides of the bivariate analysis, and then you execute the script by providing the headers for the 2 sets of traits that you'd like to correlate with, and execute the script by forwarding it to the Solar. And similar concept is for the QTL analysis script. Now, next important topic is how do you execute Solar in parallel environment. So typically imaging genetic projects involve performing genetic analysis in a very large number of traits, and therefore parallel processing becomes necessary. Unfortunately, Solar has never been designed for use on the symmetric multiprocessor systems because it's not multi-threaded, and therefore when you execute it, you will not gain any efficiency when your processor has multiple cores or your system has multiple processors. So to gain efficiency on such systems, you need to run it as multiple instances, and it's best to be used in a parallel processing environment such as SG or Sun Grid Engine. This is where you have to start planning the number of traits in your phenotype files. So it's best to accommodate the number of traits per phenotype files based on the cluster topology. So one way to go about it is to split your large projects to the number of phenotype files that equals to the number of cores on your cluster. So a project of 500,000 voxel-wise traits on a 100-core cluster, you'd basically split it into 5,000 traits per phenotype file using the list maker program. Then you create 100 processing directories on a shared disk space. In each directory, you execute run make univariate solar file script, and then submit each of the phenotype files using qsub, and just wait until it's finished. And then you can combine the results using the simple concatenation command. And there are two sets of scripts available in that directory that will simplify the submission jobs. So parallel script 1 directory contains very simple crun _ solar script, which will execute the solar script in a given directory. And crun _ job handler will do the same, but it will actually create a copy of the working directory on the individual temporary space of each server, and this will actually speed up the processing as well as reduce network utilization. Typically, if you only have 100 computers, you can do it using the shared disk space. Now, if you are fortunate enough to have a cluster that has 800 or like 8,000 processors, then it's best for you to utilize the temporary space on each individual server, because otherwise you will kill the network bandwidth and people will be very angry at you. So this script also assumes that all voxel-wise traits begin with voxel and then followed by XYZ, and the script will attempt to clean up after itself by removing unnecessary intermediate files, and will copy the results back to the original directory once the job is processed. A much more comprehensive set of scripts has been created by Anderson Winkler, and what he did is he actually modified FSL job scripts. So you can only use those scripts if you have already agreed to the FSL licensing. And one of the important features of this set of scripts is that it supports multiple queues. So the jobs can be submitted to queues of different priority, and therefore larger jobs can be submitted to queues with lower priority, which would be a very collegial way to use a public cluster. An important topic for which I was hoping that Tom Nichols is going to be here, but most unfortunately he couldn't, was correction for multiple comparisons or correction for multiple testing. So imaging genetic studies typically employ a very large number of imaging traits and obviously a very large number of genetic traits. So once you start blindly applying Bonferroni correction for all your traits, you basically gonna reject any potential finding that's less than 10 to the negative 14 in significance. So permutation-based and other correction techniques can now be, sort of something that will help us in terms of avoiding that, ridiculous requirement. And Tom Nichols have been working on it quite a bit, and he's now developing solar methodology for cluster-based inferences, including false discovery ratio and random Gaussian field theory. So what we have discovered so far is that structured genetic data sets are quite amendable to permutation-based significant analysis. So you can permute across subjects, across families, and across the genetic markers. And we tested this in TBSS skeletons because they're the most challenging, topology for multiple comparisons, given that they're neither a surface nor a 3D image. And what we did was, he used about 5,000 permutation to establish voxel-wise significance for univariate and bivariate genetic analysis, albeit John Blangero is not very happy with the way permutation for bivariate genetic analysis was performed, and it was permuted by a permutation included inside and outside of the family. And hopefully Tom will present more for the next meeting, but overall, his finding was that permutation significance level appears to be much closer to the significance level for the voxel-wise trait provided by SOLAR than the values that were provided by either Bonferroni or even FDR correction. So that was very sort of nice to hear. So it gives us hope that we won't be punished all that greatly by using multiple, large number of voxels in our analysis. So where SOLAR is heading, at least in terms of the imaging research. So it's quite clear that SOLAR provides a great foundation for genetic imaging analysis, specifically because of its flexible, MLE-based design and variance component-based analysis, because it can capture analysis in both structured and unstructured data sets, as well as it provides 20 years of research, research arsenal of genetic methods such as, age by genotype function. And it, actually it's quite efficient because most of its high performance functionality is coded in objective code that's, as fast as anything out there. So where we really need to concentrate when it comes to this is the front-end revision. And therefore, we're floating the idea of replacing TCL with a more flexible construct that would support modern technology, as well as, provide performance optimization. So the idea is to use Java instead of TCL. And I'm not saying the TCL part will actually go away. In fact, the two parts are actually going to coexist nicely side by side because, both Java and TCL can easily link to the object code stored in the C and C++ libraries. So if we were to redo the TCL part in Java, we can make it a lot more imaging analysis friendly and make it a bit more modern in the interface while leaving the TCL part completely alone. I mean, Java obviously supports three-dimensional arrays, it obviously supports nifty and gifty libraries, and more importantly, it's multi-threaded by native. It's natively multi-threaded, so it can make a more efficient use of multi-core and multi-processor servers. And particularly exciting part of this development would be the support for GPU units. Because for those of you who don't know what GPU is, it's basically a video card technology where you can buy a video card that has between 500 and 2,000 individual cores, each of them with about 10 to maybe 20 megabytes of RAM on it. And then you can submit massively parallel jobs to this system, and it will execute it, remarkably fast. So in essence, you can build, and I think Jason, the first speaker in this series, was referring to this, where you can build, a system of, 5 5,000 processors for $5,000. And given that genetic analysis are so amendable, imaging genetic analysis are so amendable to this processing architecture, that makes it very sort of, juicy target for us. And of course, where we want to go is make it compatible with LONI and GIST pipeline, specifically with GIST pipeline, because GIST pipeline actually references the data in memory and passes the data by reference, by basically a pointer to memory, rather than by saving and reading the files. And that makes it very attractive in terms of overall performance, because memory is becoming cheaper and cheaper now, and you can basically store all your data in shared memory and then access it on those access it efficiently on those modern multi-core systems. So in terms of performance optimization, the latest version is eigenvalue decomposition technique is actually wicked fast, and it becomes linearly dependent on the number of subjects rather than squarely dependent on the number of subjects. So further substantial performance gains can also be achieved by redesigning SOLAR scripts, because those SOLAR scripts were designed for both features, robustness, and stabilities. But we know that voxel-wise traits are already highly autocorrelated. So, for example, null models can be reused from voxel to voxel, and that can be used to greatly reduce computational time. Except you have to sometimes catch the convergence failures, which can occur because the two voxels, like in the voxel in the ventricle, can have a different null model than the voxel in the nearby white matter. So I think it's quite practical where we'll be able to achieve GWAS calculation rate at about 1,000 traits per second on a modern 2.5 gigahertz core. And it would take about 3 weeks of computation for doing full resolution voxel-wise GWAS study on a relatively modest 100 core cluster. Or potentially it can be done in a day using dual NVIDIA or ATI stream type GPU. So I'm hoping that there's going to be enough interest for the next year workshop, where we'll attempt to, mix theory, including, basic genetic knowledge and type of genetic contrast and type of genetic analysis, and practices where we can actually set up the demonstration and show you how to prepare your traits, create a pedigree, and perform those analyses. And of course, I'd like to acknowledge Laura Almazy and David Glan for their help in setting up this workshop, and NIH for funding SOLAR. Yes?
I'm still trying to get my head around this, so excuse me if I missed a totally important point there, but if I have GWAS data and I have just a few traits, I don't think I can explain. If I have just a few genes and imaging data, I would just plug, like if I'm interested in the freeway interaction with a few genes, I can just put it FSL. So the application I see for that one is that I have GWAS data and imaging data, and I want to try to make sense of that.
In a sense, yes.
But then I'm stuck with files I want to chop down. I have to chop down my genetic data, and I have to chop down my imaging data, and I have to paste it all back together, and then I don't get, or what do I get out in the end? An image?
You do eventually get
Giant table window.
I'm not saying that it is easy right now. I'm not saying, but
I mean, I'm just asking what is
Could you?
Size, like the striking point is the best on solar compared to clean or just a normal imaging model.
Well, okay, so the point link is that once you have, it's great for the case, case control type studies, right? Once you have a related individual in that mix, now you can't really use it anymore. So that creates a bit of a problem, right? So if you have relatives that are included in the part of your data set that sort of controls your, sort of curve goal. So, one can actually argue that why would one even use Blink when, he or she can code, logistic regression in a C++ code in half an hour or so by.
First of all, because I have to chop down my GWAS data.
Well, you can just read your GWAS data from, write a small C++ program that will actually hit your.
Or I can just put my thing in.
So I think actually, not to be offensive to either of you, but this is a lovely conversation. Probably best served with beer, but yeah. So the issue is as follows. In one portion of today's schedule, it says that this seminar continues until 6:30. In another, it says that it continues until 6. I'm sort of feeling like it should have stopped at about 5:30. That was about 5 minutes ago. Except I'm the last speaker, so that's kind of bad. So what we're going to do is as follows. Yeah.
I think the bus.
Right. The bus to the hotel is at 6. Basically, I'm just going to push through in about 20 minutes and finish up the kinds of things that I was going to do, which means that inevitably what I will say will be confusing and probably bizarre. But if you have any questions about it, find me. The slides will be available as well. Really, my job was to show you some examples of some of the things that we've been doing kind of more in detail, just to give you a feeling for what's going on. Many of them have been published, so they're fairly straightforward. You can find the references at different places. At some points, I show things that aren't necessarily done in SOLAR, but are using similar sorts of models, just to give you a sense of how that would work. And then finally, I was charged with giving you examples of what we're referring to as an Irvine analysis. That will probably be the thing that gets cut the fastest, and that I may kind of fly through. But we'll go with what we have. So I was told to give examples of heritability, genetic correlation, linkage, and GWAS. So here we go. Heritability first. So thinking about it again, heritability is an estimate of between 0 and 1, where 0 would indicate that genetic factors don't really influence a trait, and 1 where they're suggesting that it's under complete genetic control. The biggest issue here is that stronger heritability doesn't imply a simpler genetic. It just implies that it's stronger heritability. And so people spend a lot of time thinking about it in that way. So here are a couple of examples of things one could do with something like SOLAR if you had family data, or MX if you had twin data, or various other types of software. These are 486 individuals from pedigrees that we ran through FreeSurfer, and we then derived cortical thickness measures, which is what I'm showing you here, and surface area measures, which I'm showing you here. We derived each one for each gyrus, determined their heritability, and the color scores here are actually reflecting the heritability, right? So you can generate an image in the same way. We also did this before I knew that Peter was working on routines to do so. So it is possible to go in, run a voxel level. In this case, each of these is vertices, and the heritability estimate is telling you the heritability we got for that vertex based on a T1 weighted image in the cortical thickness, right? So I'm showing you heritability estimates. So in this case, it's ranging between 0 and 0.75. The images look a little bit weird, primarily because I screwed them up when I stretched them a little bit, and I apologize for that. But the point being that this particular image could be replaced with any statistic that you output from SOLAR or from any statistical package. The question is, once you get it into a space like this, in this case a surface, are there then subsequent statistics that you're going to want to do in order to be able to tell you what seems to be heritable and what doesn't? Right? Now, one of the big points that we have here is that almost everything in cortex is actually heritable at some level. So then we might want to make inferences about what areas seem to be more or less heritable, or what areas seem to fall together in something like a multivariate analysis. You could also, quite simply, do the same thing, except now each statistic is representing an association analysis. And as we've also, of course, shown heritability estimates for DTI measures and heritability estimates for resting state fMRI based on using ICA methodologies from the FSL group. The point really that's important here is that we know that basic anatomic and DTI and functional imaging data have reasonable heritability associated with them. We're getting to that point. When you get to task-based fMRI studies, there are a couple of papers out there that have done heritability estimates, but not all of them. So last year, there was a good discussion about whether or not you actually ever needed a heritability estimate, and why would you need that? I would make the argument that until we have all become extremely comfortable with the idea that something is heritable, we need to be able to show that it is influenced by genetics before we make that assumption. Okay, the limits of heritability, because there's some issues here with the inferences that we make. Heritability is a population level parameter. So it's summarizing the strength of genetic influence on variation in a trait. It doesn't tell you anything about a particular individual. It doesn't tell you anything about how complex the underlying genetics of that trait might be. It's basically letting you in the door to do subsequently what I believe are somewhat more interesting genetic studies. Heritability is the aggregate of effects of multiple genes, as was discussed previously, and so I don't think we need to get into that. But what this means is, if you go to make inference about something like this, where I'm saying to you, the heritability of the default mode is about 0.42, that's telling us that some level of the physiology associated with default mode processing is genetically mediated. I don't know how much further I would really push that, though we did do additional things in this manuscript. Okay, genetic correlation. There's been a lot of discussion of genetic correlation or of multivariate analyses. In SOLAR discussions, what you're really trying to measure here is pleiotropy. Sometimes individuals particularly individuals who work with twin data, get very uncomfortable if you use the word pleiotropy in this context. But in essence, what you're doing is you're trying to find the overlap. A genetic correlation is just like any other correlation. It varies between -1 and 1, 0 meaning that there is no effect, -1 or 1 meaning that it's completely associated. So I thought actually, so sometimes one's memory plays tricks on them, and I put this slide in here, and I can't remember if this slide is from my paper or from Eric Schmidt's paper. So good, so it was from mine.
Much better quality.
In your paper, undoubtedly. But basically, what it's showing is an effect that we wanted to look for. And this is a known one for people who do this type of image analysis. It's looking at the relationship between surface area on cortex and the thickness of that cortex. And the phenotypic correlation between surface area and cortex is very, very, very small or non-existent. Whereas for gray matter and cortical thickness and gray matter and surface area, there is some relationship. And indeed, these are actually these are actually the, oops, sorry about that. These are actually looking at the genetic correlations of these groups. Or actually, these are the phenotypic, but the genetic follow the same thing, indicating that we didn't find a genetic correlation between surface area and cortical thickness. So the inference that you would make from that is that different genetic factors are influencing the area of cortex versus the thickness of that cortex, right? That's the inference that we're drawn from, given Danny Weinberger's caveats that there's a lot of measurement error or variance that we might want to think about. But nonetheless, that would be the inference you would make from this. There was another paper that we did which was much more basic, which basically said, okay, we want to look at the FA values in the superior longitudinal fasciculus and performance on a delay response task that was shown to be associated with schizophrenia. Now, the reason we were doing this was because in imaging studies we've shown that the bold signal associated with doing this particular task shows you brain regions around the superior longitudinal fasciculus, and that there's a correlation between that bold signal and the FA values in the superior longitudinal fasciculus. That was in one sample. Here, what we wanted to do was to show whether or not there was some genetic relationship, and indeed there was, where there wasn't a genetic relationship in some of the other things. The problem with this, which was pointed out to me by a schizophrenia genetics individual who really doesn't do imaging, is that this makes the story a lot more complicated, right? Because if you're choosing one of these traits as your intermediate phenotype, you make the assumption that there's all some strong relationship. And while there is a relationship, it's not, our genetic correlation is 0.59, which is reasonable, but it's not perfect. And so if you're doing this and you don't think about these kinds of intermediate phenotypes as much, or into phenotypes, you realize that there's going to be a complexity here as well. Okay, QTL localization linkage. So I just threw up an image from Peter Kochenov's paper. This is actually a very interesting scenario because, as Peter pointed out, doing a bivariate analysis is sometimes quite useful, and that's actually what was done here. What you're seeing here are is the LOD score at 3.82 for bivariate linkage using whole brain white matter hyperintensities and blood pressure, right? So blood pressure is heritable at some level. White matter hyperintensities were heritable at some level. There is a genetic correlation between the two of them. And while you didn't get a significant LOD score with either trait independently, together you did in this location. And that's really the power of the things that Peter, I think, was trying to get across, is that sometimes when you have traits that show some underlying common genetic features, focusing in on those can give you power that you might not see otherwise. A QTL localization for an association measure. I start out by showing you Jason Stein's earlier paper from NeuroImage, where they actually ran a GWAS analysis on each and every voxel in a VBM study. I don't remember exactly how long this took, but it took. Oh, he did downsample, but it took a while. This is the kind of study where I don't really take the results of the study as being what's interesting about it. I take the fact that somebody sat down to do it, right? Because I have a sense that in time we will be doing stuff like this. But there's a lot of methodology that needs to be worked out before we can really draw inference from it, I believe, because you have a problem of testing 500,000 SNPs and 500,000 voxels. So figuring out exactly what an appropriate way of drawing inference for that would be, I think, is important, right? But the methodology and the idea of doing so is moving along. And as a community, these are the kinds of things I think we're going to have to be thinking about, and the kinds of things that we're going to have to really be able to model well. Okay, on a much more simplistic point, we recently delineated the volume of the amygdala and ran a GWAS analysis with it and came up with a genome-wide significant hit on the exon of FGF14, which is a human growth factor hormone. We can talk about that at some point if you want. The manuscript is actually still under some level of preparation. Okay. The final point was endophenotype ranking. This is really the idea that endophenotypes, you can really think of that subset of biomarkers that are heritable and that are associated with a trait. Laura yesterday went through a number of these criterion. The basic idea, as explained by Bob Builder, is that you want to go somewhere from the genome level, I don't know, from the genome all the way up to the syndrome, and you have all of these intermediate points. And as he states it, what you really want to do is measure some of these things, and any of these things could be conceptualized at some level as an endophenotype. And then what you do is you make a multi-level hypothesis. The problem is I'm not really smart enough to make a multi-level hypothesis, so I kind of think that they're a little bit unconstrained in many cases, right? So we wanted to come up with some way that we could evaluate potential endophenotypes and discover new ones if we didn't have a specific hypothesis. By so doing, we decided that an endophenotype just needed to be heritable and show some genetic correlation with the trait that you're interested in. And if you can show those two things, you could create a ranking value. In this case, the endophenotype ranking value, which we named IRV after this guy, Irving Gottesman, who coined the term endophenotype. Actually, the first time we discussed this in public, it was Laura Almasy who gave a talk on this at a psychiatric genetics meeting. And Irv came to me later after the talk with a huge smile and told me it was the best 80th birthday present he ever got. I didn't remind him that actually I believe his son had had a child about 3 months before, but we didn't need to talk about that. At any rate, it's a reasonably straightforward measure. It is basically deriving the standardized genetic covariance between two traits. You derive the standardized genetic covariance by multiplying the square root of the heritability of trait 1, the square root of heritability of trait 2, and their genetic correlation. The reason you do this is that if you have data where you can generate these types of data, you can easily rank the measures that you need. All right. I think all I'm going to do is say to you that we ran this on a bunch of cognitive data. We ran this on a bunch of imaging data. And we ran it on a bunch of transcripts. And then we did a bivariate genetic analysis with it and came up with a LOD that got over our 3.5 for depression, which is useful because, as we heard yesterday, 20,000 individuals in a sample of in a depression GWAS study came up with nothing. So this approach gave us something, at least quasi something. Okay, that particular locus also explained variation in recurrent MDD, which was our general phenotype, the expression of RNF123, the volume of the ventral diencephalon, but not of the BDI score. Okay, thank you. You have 8 minutes until the bus. Yes?
Can I give a 30-second answer to the question previously about what does solar do? What does solar do well? The answer is,
That sounds too hard.
no, it's the right question. The answer is large families. If you have unrelated individuals, there are probably other programs that do what you want to do that will be more convenient for you.